Hello everyone, this is Hemsankar Lauge. I'm excited to present my research on the topic engineering a e-surface display platform for peptide MSC expression and rapid characterization of T-cell receptor mimic TCRm antibodies. As we know, our immune system uses T cells to detect and destroy abnormal cells. These T cells recognize tiny fragments of proteins called peptides that are presented on the cell surface by a special molecule called major histocompatibility complex (MSC). When this process goes wrong, like in case of cancer, our immune system may fail to detect tumor cells. And to fix this, scientists have created special antibodies called T-cell receptor mimic TCRm antibodies, which can see the peptide MSC complexes just like natural T cells do, and making them like a powerful tool for cancer therapy. However, making and storing these peptide MSC complexes in the lab is really hard. They are unstable and don't always fold properly outside the body. That's where my project comes in. So we wanted to solve this problem by building a reliable platform for displaying these peptide MSC complexes. And as you see in this figure one, it's like e-surface display as a platform for peptide MSC expression. So for this project, we use a technique called e-surface display, which allows us to display proteins on the surface of e-cells. And we combine this e-surface display technique with a single chain trimer SCT. So SCT basically stitches together three main parts like the peptide, beta 2 microglobulin, and the MSC heavy chain into one continuous protein. And this way, like everything folds together more reliably and stays stable on the e-surface. And for this project, we used a specific strain of E-cell called EBY100 to display a tumor-associated antigen called MAT1 that is presented by a human MSC molecule called HLA2. Now, let's discuss how we did and how we validated it. So for this, like we first like grow these E cells into in a selective media, and then we introduced our SCT gene into the E cells using electroporation technique, and then allowed them to grow and express the protein on their cell surface. And to check if our system worked or not, we used flow cytometry technique. Flow cytometry is a technique that uses lasers to detect fluorescent signals. And for flow cytometry, we stain the E cells with antibodies that bind to HLA2 and beta 2 microglobulin. And as you see in the experimental results section, for the transfected cells, we saw like the high level of surface expression of MAT1 and HLA2 peptide MSC complex. But in case of non-transfected cell, which is our like the control, we didn't get any like the source level of high surface expression of MAT1 HLA2 peptide MSC complex. So this tells us that this MAT1 HLA2 peptide MSC complex was successfully expressed on transfected yeast cells. This rightward shows us this geometric mean fluorescence intensity, and in this case, we saw like the high level of like the GMFI in case of transfected, but with case of non-transfected, there was no such like the fluorescence intensity saved. And the bottom graph is just like the histogram, and which further clarifies that MAT1 HLA2 peptide MSC complex was successfully expressed on the surface of the yeast cells. So now, like we can say that this yeast-based system gives us like the cheap, stable, and reproducible platform to screen T-cell receptor mimic antibodies. And instead of growing like expensive tumor cells or working with fragile proteins in solution, we can now use yeast cells like tiny display boards to show different peptide MSC targets. And as a future direction, we plan to now expand this platform by generating a library of peptide MSC that could present diverse tumor-associated antigens, and this will allow us to further develop and characterize additional T-cell receptor mimic antibodies. And also, one of our main future direction is to begin screening T-cell receptor mimic antibodies that selectively bind to the SMART1 HLA2 peptide MSC complex. And we believe this platform could help accelerate the discovery of new cancer-fighting antibodies, especially against tumors that present very few targets on their cell surface. Thanks for listening, and a huge thanks to Imprint for supporting this research. Thank you very much.